now, and that will go through the Q&A session at the end of the presentation. Dr. McKay, the floor is yours.
Thanks, Patrick. So hi, everybody, and welcome to our Monday seminar this spring. Our seminar series is co-sponsored by, in addition to the School of Public and Community Health Sciences, also the Global Public Health minor, with the support of the Institute of Health and Humanities as well. And so thrilled to welcome one of our Global Public Health minor steering committee members, Professor Angie Luis, who's going to be speaking this week on her research having to do with how biodiversity affects risk of zoonotic disease. So I'm going to do a little intro and then turn things over to her. So Dr. Luis is originally from Oklahoma, where she was not taught evolution in public schools. And so to be rebellious, she did some reading on her own and found evolution to be utterly fascinating and fell in love with biology. She received her BS in zoology from the University of Oklahoma and then worked in an immunology lab for 5 years. Although the interactions between microbes and their hosts was interesting, she found her love with the bigger picture of disease ecology, how diseases spread through populations. She received her PhD in ecology from Penn State University in 2010, and then did postdoctoral research at Colorado State University and Princeton University before joining the faculty at UM in 2014. Here at UM, she studies how infectious diseases spread through populations, which leads to outbreaks, and ways to predict and control them. So in her free time, Angie enjoys the great outdoors of Montana, including hiking, climbing, and snowboarding, and experiencing life through the eyes of her 4-year-old. And I can't think of a more interesting or appropriate topic for us in these times of pandemic and disease outbreak than your talk, Angie. And again, I'm so thrilled to have a steering committee member join us from GPH in our speaker series this year. So I'm going to turn things over to you and ask you to take it away.
All right, thanks so much, Kimber. So I'm going to tell you today about how biodiversity affects risk of zoonotic disease, mainly using a case study of what I study, Hantavirus in the US specifically. So as you know, today biodiversity is being lost at an alarming rate. Many agree we have entered the 6th mass extinction. Species are being lost exponentially. So here is a graph where we're looking at over time, the cumulative extinction as a % of those evaluated by the IUCN. And we see that especially over the last 100 years, extinctions have increased dramatically over what we would expect based on background rates from the fossil record. At the same time, we're seeing infectious diseases emerge at an unprecedented rate. So this figure is showing since the 1980s, the number of outbreaks that have occurred in humans. And you can see they've increased dramatically, especially those that are zoonoses. So those are things that are transmitted from animals to humans. And of course, right now we're living through a pandemic of zoonotic origin. So a big question right now is, are these two things related? We're losing biodiversity, we're seeing an increase in outbreaks. Is one related to another? Or more generally, how does species diversity affect infectious disease dynamics? This is a big question in my field right now. And there's a couple of possibilities and a couple of patterns that we do see. So one way that these two things might be related is through what we call the dilution effect. And the idea is that as biodiversity or species diversity increases, it dilutes out infection. So we see a decrease in infection prevalence or risk of spillover. And I'll go through how that might work in a second. What we call this dilution, because species diversity is diluting out disease. If we're thinking of the patterns that we've been seeing, we're losing diversity, we're moving in this direction. So as we lose biodiversity, we're seeing an increase in prevalence. So how might this work? One example is Lyme disease. So for this infection, ticks are needed to transmit the infection between individuals. White-footed mice are the main reservoir host. And so ticks come along, they feed on infected mice, here shown in green, and that's how they pick up the infection. And now they can go on and bite a human and transmit the disease. if we have more mammals in the system, if we have higher diversity of mammals in the system, then we've got a lot more things that the ticks can feed on. And a lot of these other hosts, like possums in particular, when they try to feed on possums, that dilutes infection because those possums are not reservoir for the bacteria. The bacteria doesn't grow well in the possum, and possums can even eat the ticks and help decrease the tick population. And so that leads to lower prevalence in the ticks and then less of a chance of transmission to humans. So here's an example where diversity is diluting out disease. So that's an example of this dilution effect. And there's this big debate right now about how prominent this dilution effect is in nature. We know it happens for Lyme disease, but how many other systems does this happen in? What conditions are necessary to create this dilution effect is something which is kind of a big hot topic right now in my field. Evidence is mounting that there's a number of systems for which we see this, but actually we could see the opposite. There are some systems where we see this opposite effect, that as you see increased diversity, you see an increase in infection prevalence. So how could that work? So one way this could work is you have areas of really high biodiversity, then you could also have a high pathogen diversity just within those species. And so you get a greater chance of transmission between species, greater chance that some of those pathogens are going to be able to be transmitted to humans. So higher zoonotic pathogen diversity, greater chance that they actually get into humans. And so how does this relate back to what I was telling you about increased infectious disease as we're seeing biodiversity loss? It might seem counterintuitive, but that could also lead to this pattern. And so one way that could work is if it's not the biodiversity loss itself that's leading to outbreaks, but things that we're doing to cause the biodiversity loss. For example, if people are going into these really biodiverse areas and doing the things which cause biodiversity loss, like destroying habitat, wildlife trade, those things which make it more likely to come into contact with these diseases and get spread to humans. And some of my own work suggests this could be the case with bats. So here is a map of bat diversity across the globe. So the number of bat species. So there's some areas in South America and Southeast Asia, for example, where there's up to 50 bat species in one area. And we see that in those areas that have particularly high bat diversity also have a lot of zoonotic viruses in bats, things which could get transmitted to humans. And it seems like there's a lot of transmission occurring between species. So bats not only have more viruses total, but they're transmitting to each other more. And this could be why we saw, for example, a Nipah virus emerge. So in 1999, there was a deadly outbreak of a newly identified virus in Malaysia. If you've seen the movie Contagion in 2011 with Jude Law and Kate Winslet, that was based on the Nipah virus. It led to 250 cases with 103 deaths. The reservoir was a fruit-eating bat species. So basically what happened is people moved into these really biodiverse areas. They cut down the native fruit trees these bats would roost in, and instead they put in pig farms adjacent to fruit orchards. So now bats didn't have their native trees. They're now roosting in these fruit trees and picking up the fruit, half eating it, dropping it to the ground. Now there's fruit with virus on it, and pigs are coming along and eating that contaminated fruit and getting infected and spreading that to humans. And so here's an example where we went into really biodiverse areas, did the things which cause biodiversity loss, cut down a bunch of habitat, put up our own agricultural practices, and then led to a greater chance of spillover between these viruses and domestic animals and humans. This, we can't say for sure exactly where SARS-CoV-2 came from, where COVID-19 started, but there's evidence is mounting that the spillover did first occur in a seafood wet market in Wuhan, China. And these wet markets are places where there's close contact between different animal species and people. And that increases the chance of transmission between all these different animal species and between animals and humans. And so here's a picture. This isn't from that particular market. And these are avian species, but we know that transmission has occurred, zoonotic transmission has occurred in these types of wet markets before. So they're examples of 2 different bird flus that were likely originated in these types of wet markets in the past. So basically you just have this area where you're bringing a whole bunch of wildlife together in this place where here this poop is dropping onto the animals below. It's a great chance for transmission to occur that wouldn't normally occur. So we're taking animals from these biodiverse areas, bringing them together in artificial environments and creating this great place where animals can transmit between each other and then transmit to humans. So for cultural reasons in the region, people want to see the specific animals that they're buying. They want to see them slaughtered in front of them so they know what kind of meat they're getting. And so that slaughtering also creates a great chance for things to become aerosolized and for transmission to occur. So where did SARS-CoV-2 come from? So this coronavirus was very closely related to a previous coronavirus, SARS-CoV-1, 80% identity to SARS-CoV-1 and 50% to MERS-CoV. So both of these, we've identified that bats are the reservoir host for them. And then we found that there is a 96% match between this coronavirus and horseshoe bats in Southwest China. And these horseshoe bats are really cool. They're really interesting bats that have these things on their faces called nose leaves. We think that they help with echolocation. They help to focus the sound. So we haven't identified any live bats that were at this market, but it's likely that the virus spilled over from these bats into a reservoir, into an intermediate host that was there in these markets, and that led to spillover into humans. So back to the biodiversity question, how does this relate to our question of biodiversity? This is one of those regions where there's really high, there's very high diversity in the bat species and a high diversity of zoonotic viruses within those bats. And these things which lead to biodiversity loss, like going in and cutting down habitat or bringing wildlife close together, those things are potentially what's leading to these spillover events rather than the biodiversity loss itself. So in summary, we could see that species diversity, as you have species diversity, that it dilutes out infection, like with that example from Lyme disease, or we could see the opposite, that areas with higher biodiversity have higher infection prevalence, and that this amplification effect is actually more prevalent. So this is a pretty big hot topic. There's been a big debate in my field over which one of these things seems to be more common in nature. When would you expect to see the dilution effect versus the amplification effect? So that's something that I'm really interested in. And a lot of the work that's been done up to this point has been on vector-borne disease. So things like Lyme disease needs a tick to transmit between different hosts. But the majority of infectious diseases don't need a vector like a tick or a mosquito, but are transmitted directly, like this coronavirus is, directly from person to person or between animals. So if we're interested in a directly transmitted disease with one main reservoir host, this SIR framework is useful to help understand the mechanisms of disease spread. And so with this framework, individuals are grouped into different categories with respect to their status. So we have susceptible individuals. So they're uninfected but could get infected. Or we have infected individuals and they're contagious and can spread it to others. Or we have recovered individuals that got over infection and now they're immune and can't get it again. And so over the course of an outbreak, individuals will move through these different categories. So everyone will start off as susceptible, but they can become infected if they interact with an infected individual and move into this I class. And then they spend some time in the I class when they're infectious and contagious and can spread it to others. And then when they recover, they move into the R class. So we can categorize, we can describe how this occurs. And one of the important mechanisms is this transmission rate, the rate at which susceptibles can become infected after interacting with infected individuals. So the main things that lead to transmission, the mechanisms that are important for transmission, and the things that we need to look at if we care about infection prevalence are host density. How many susceptibles are there that could get infected? The contact rates between the susceptibles and infecteds, and then how contagious a particular pathogen is. And so this contact rates, and this is the same thing as sort of contagiousness, the probability of transmission given contact, which I'll call transmissibility. So these two things we often refer to with this beta transmission rate. So the main things that matter if you're interested in how does biodiversity affect infectious disease, you need to ask, how does biodiversity affect host density? How does biodiversity affect contact rates between individuals? How does biodiversity affect transmissibility or contagiousness? And so that's what I'm asking with my system. We would expect dilution, that biodiversity is diluting out disease, if species diversity leads to lower host density, lower contact rates between individuals, or lower transmissibility. We would expect the opposite. We would expect an amplification effect if species diversity leads to higher host density, higher contact rates, or higher transmissibility. So as an outline for the rest of my talk, first I'm going to tell you about my Hantavirus system, where in the Southwest US, we do see a dilution effect. And then I'm going to ask, how could this dilution effect, what's the mechanism for this dilution effect? Could it be host density? And so I'm going to spend some time looking at this potential possibility that it's because of host density. Then I'm going to look at all three of the possibilities for dilution together. And then I'm going to end with a sort of broader view of all the different ways you could get dilution or amplification. So a bit about my system. In 1993, in the Four Corners region of the Southwestern US, 13 people died mysteriously after acute respiratory distress, and another 14 would die within the year. And the cause was determined to be a previously unknown hantavirus that was termed Sinombre virus, or SNV. So this causes hantavirus pulmonary syndrome in humans. And it's really nasty. 1 in 3 die after being diagnosed with HPS. There have only been about 750 or so cases in the US, so it's pretty rare, but it is quite deadly. So here's a distribution of human cases. I think this was as of like 2 years ago or so. So you can see it's mainly a problem of the western half of the US. The main reservoir is the deer mouse, Paramiscus maniculatus. And humans are spillover hosts. They get infected when they come into contact with excrement from these infected rodents. So for example, sweeping up mouse poop, it becomes aerosolized and they breathe it in. Hantaviruses weren't new to science at that point, but just new to the Americas. So all of these blue over here have been discovered since that initial outbreak in '93. But we knew about hantaviruses before because of some infections that occur in Europe and Asia. Those infections tend to cause what we call hemorrhagic fever with renal syndrome. It's a bit less deadly of a disease. Some of the cases are less than 1%, and they can go up to 10% fatality. Whereas these cases over here in blue, the hantavirus pulmonary syndrome, can be 35–40% mortality rates. But we knew about hantaviruses. We knew that they tended to have rodent hosts. And so when we discovered this thing, when it was sequenced, we discovered it looked really closely to another hantavirus. We started looking in rodents and found, yes, this is the rodent host, this deer mouse. So how does this relate to that biodiversity question? So my colleague, Jim Mills, he retired from the CDC, but this is one of the figures that he published in 2005, showing this really strong dilution effect. So the Simpson's diversity index is showing the diversity of small mammals at a site. Sites which had a higher diversity of small mammals like a lot of chipmunks, different types of mice, other rodents, tended to have lower prevalence of hantavirus in deer mice. But sites that had lower diversity had higher prevalence. And so this is a pretty strong dilution effect. So in the Southwest, we're seeing that increased small mammal diversity leads to decreased hantavirus prevalence in deer mice. And so that's a dilution effect. So first, what's the mechanism for this dilution effect? Is it lower host density? Is it lower contact rates between deer mice? Or is it lower transmissibility? And remember, these things are incorporated in the transmission rate. So first, I'm going to look at the possibility of host density. So we have deer mice that are part of a population. And let's just say these circles represent their range of contacts. Maybe it's like their home range. And so everywhere that these intersect, this mouse is in contact with this mouse because its ranges intersect. So let's say we have some deer mice and 3 of them are infected, represented by this green. And so each of these greens could transmit to anyone they're in contact with. And let's say after, let's say the next month, now everyone in green has infected now everyone that's in red. You notice this one was in contact with this one, it overlapped, but it didn't get infected because the probability of transmission given contact isn't 100%. That's that transmissibility measure. So let's say this is our sort of baseline scenario where we have this deer mouse population and a month later we have 3 new infections. So let's think about how the dilution effect could work here. So let's say the dilution effect is occurring through decreased host density. So we've got our original community, we add some diversity to it. Let's say we add some kangaroo rats, some chipmunks, some prairie voles, and now we have a more diverse small mammal community. And there's a lot of diet overlap between these individuals, between these different species. So there appears to be pretty strong competition with some of these species. And so that leads to decreased host density. So fewer deer mice are around because we have more competitors around. And so when we remove deer mice from the system, we now have lower host density and now a lower chance of infection. So now in this scenario where we've increased diversity, now we've only got 1 new infection when before we had 3. So there's an example of increasing diversity leading to decreased host density and decreased transmission. But what about the other side? What about contact rates? Let's say it's due to changes in behavior. So here's our mouse population. Now we add some diversity. One thing that might happen is that animals change their behavior. These are dominant competitors, and maybe animals shrink their home ranges. I don't know if you could tell what just happened there, but the circles got a bit smaller. And so because they're around these competitors, they might change their behavior to where they're not moving around as much. And so now deer mice aren't contacting each other as much. And now that leads to decreased number of infections. And so we went from 3 down to 1. The other possibility is that could happen through decreased transmissibility. And so let's say here's our community, we've added diversity. And so now, even though these were in contact with each other, they didn't get infected because the probability of transmission given contact or transmissibility went down. It's harder to come up with a good scenario for this, but maybe it's because some of these were mutualisms with our mice. And so deer mice do better, they're healthier when they're in the presence of some of these other species, and that could make their immune system stronger and less likely to get infected if exposed. So these are just some hypothetical scenarios. So let's ask, what's the evidence for these 3 different mechanisms? Well, first, let's look at host density. So in order for host density to be the mechanism, we need to see that increased prevalence with increased density. So for example, when I say prevalence, I mean the proportion of the population infected. So what we would need to see is that as deer mouse populations increase, the prevalence of infection increases. And so if you saw increased diversity, then that would lead things going in this direction and would lead to a decrease in prevalence. So we call this density dependent transmission. If as density increases, prevalence increases. And this is true for a lot of directly transmitted diseases. If you have a more dense population, you're going to come into contact more. But there are some diseases for which we don't see that pattern, for which we see what we call frequency dependent transmission. And so in those instances, as you increase host density, it doesn't increase the prevalence of infection. This is more common for like sexually transmitted diseases, for example. So if you're in a population, if you move to a more dense population, doesn't necessarily mean you have more sexual partners. But if there's more direct contact, then that typically does increase prevalence. So we need to show for this disease, do we have density dependent transmission? Then density could be the mechanism here. If not, if we have frequency dependent transmission for this infection, then host density can't be the driver, and we can rule it out. So we have a lot of great data to answer this question. My colleagues in Butte at Montana Tech have been collecting data from Montana for more than 20 years. And Jim's time at the CDC, he oversaw a bunch of different data collection sites in the Southwest from 1994 to 2006 at all these different sites. And so here I'm showing for each of these, for a handful of these sites, blue are Montana sites, green are Southwest. I'm showing how mouse density, deer mouse density, is related to hantavirus prevalence in deer mice. So remember, we're looking for a positive relationship between density and prevalence. I don't see it. None of these sites have a significant positive relationship. And in fact, there's a number of sites have a significant negative relationship. Grand Canyon, Zuni, Cascade, in particular, Gold Creek. A lot of sites have a negative relationship, which doesn't really fit our understanding at all. So what's going on there? Well, previous studies that have looked at this have just said, okay, density can't be the driver of this relationship because there's not a positive relationship between density and prevalence. But I want to dig more into that and see what's really going on there, because what we're seeing doesn't make any sense. So I'm going to do that by zooming in on one particular field site, Cascade County, Montana. So this is one of those sites for which there was a negative relationship between density and prevalence. So what the data look like, there's 15 years of monthly live trapping data. They went out even when there was tons of snow on the ground every month, live trapping animals, putting ear tags on them, tracking them through time, taking blood samples, looking for antibodies to look for infection. So here's what the data look like. So total mouse density over time is in black. And number of infected mice is in red. And you can see it's highly variable over time. There was a significant kind of increase in the mouse population over 2002, 2003, and then a decline. And then there was a subsequent outbreak around that time within the mice. Seroprevalence ranged from 0 to 30%. So again, this was one of the sites that had a significant negative relationship between density and prevalence. And so we're trying to figure out what's going on there. And just looking at correlations isn't telling us much. So I'm going to explore this question with one of those SIR type models that I told you about. So SNV in deer mice is a chronic infection. Once mice are infected, they're infected for life. So there's no R class. So that makes things simpler. So I'm going to use an SIR model or SI model, where I'm going to look at how susceptibles become infected, try to estimate what this transmission rate might be. But we also know, and I'm going to try to figure out, is there density or frequency dependent transmission? We need to incorporate the disease-induced mortality rate. My work shows that mice do sometimes die from this infection. It's not nearly as deadly as it is to humans, but it does decrease their survival. And it's really important here to include population dynamics, so birth and deaths of the deer mice and how they're changing through time, because we saw deer mouse populations are moving up and down a lot, so we need to account for that. And so we're going to do that using a carrying capacity, which is changing through time. And so these birth and death rates are density dependent and depend on where the population density is in relation to the carrying capacity. And so I love talking about the math, but I will spare you and just show you that there is math behind all of this. And in particular, we can incorporate the two different functions of density dependent versus frequency dependent transmission and ask which model fits better. Are things density dependent, or does prevalence not depend on density? So I'll show you how the model fit. So here's the infected numbers of mice over time. And here's our fit from our density dependent model. It does a good job at predicting the kind of major outbreak. It kind of missed some of the smaller outbreaks, but we're talking about just a few infected individuals. So how did frequency dependent transmission fit? Yeah, it didn't fit at all. It was a terrible fit. There was really strong evidence that the density dependent transmission fit better. And I've applied this to other data sets and shown the same thing, that density dependent transmission, when there's enough data to discriminate between the two, it always does better. But I told you before that what we should see is that as density increases, we should see an increase in infection prevalence. But this is what we saw in the data, and these two things don't match up. So why not? Well, the reason they don't match up is because this is what we would expect. This increasing relationship is what we would expect if the system was at equilibrium. And so what I mean by that is if we had a mouse population density of 20 and it stayed there, then prevalence should be about 20%. If we have a mouse population density of about 40, but it's unchanging, it stays there, it should be about 60%. But the problem is this system is never at equilibrium. The density is constantly changing. It goes from 20 to 60 within a few months. It's constantly moving up and down. And so that means that prevalence is constantly sort of lagging behind the density. It takes time for the infection to spread and for prevalence to increase after density increases. And so the prevalence is lagging behind the density. And so what does it actually look like when we compare using the model? It looks very similar to what we actually see in the data. So this is what we see if things are at equilibrium, but if we actually let the carrying capacity change through time as it did, we see an overall negative relationship. So what does that mean? That means that transmission is density dependent in the system. So prevalence does increase with density, but there's a time lag. It's lagging behind. And I don't have time to tell you about all the other cool things that go along with that. But what I just showed you was that host density could be the driver for this dilution effect. Other people have discounted it in the past, but we're showing here that it's possible. But I haven't actually showed you that it is the driver of the dilution effect yet. So now we need to evaluate, is this dilution effect driven by host density, or is it one of these other things? Is it contact rates or transmissibility? So we're going to look at that now. So remember, these two things are part of the transmission rate, and I estimated the transmission rate in my modeling exercise above. And so I can do that again for all of the different sites for which there's a range of host diversities. So what we see, especially in the Southwest, there's a big range of diversity at these sites. Some of them are really diverse, like Civietta has a really high small mammal diversity. Some of these other sites have lower small mammal diversity. And so I can take each site and say, what's the transmission rate for this site versus this site versus this site, and see how that relates to the diversity at each site. So I'm going to fit my model to each site separately, estimate a transmission rate for each site, and then compare that to diversity for each site. And so if host density is driving the dilution effect, we'll call that hypothesis 1, then what we would expect to see is that all the transmission rates aren't changing with species diversity. It's just being driven by density. But if it's being driven by lower contact rates or transmissibility, which is part of that transmission rate beta, then we would expect that beta will vary with diversity. At more diverse sites, we'll see a lower transmission rate. So I can do that and fit the model to all these different sites. And so here's just showing you some of the model fits. Again, the blue are Montana sites. So this is near Polson, Gregson. Here's some Southwest sites, Grand Canyon sites, some Colorado sites. So each of these, the red is the observed number of infected individuals, and the purple dashed line is my model fit. And so it's showing the model's doing a good job at fitting at all these different sites. And the model is driven by host density. So the model that takes into account the lagged effects of host density on prevalence fits the data really well. So what about transmission rates? So again, what we would expect to see if host density is driving this relationship, then we're not going to see a change in the transmission rates with small mammal diversity. If lower contact rates or transmissibility is leading to this dilution effect, we would expect transmission rate beta to decrease as small mammal diversity is increasing. So what do we actually see? We saw the opposite. So what does that mean? So transmission rate is higher with higher species diversity. So basically what that means is that host density is driving the dilution effect. When we have higher species diversity, we're seeing fewer deer mice, and that's leading to less infection. But at the same time, we're seeing what I've called a component amplification effect. Basically, the idea is that one of these things, or both of these things, contact rates or transmissibility, is actually increasing with higher small mammal diversity. But the effect here is just stronger. So what we're seeing is that both dilution and amplification are occurring in the system. The dilution effect is just much stronger. And so it's the dominant relationship that we see. But amplification is also happening at the same time, which is really cool. No one has ever shown that before. So the dilution effect is mainly driven by mouse density. Increased diversity is leading to decreased mouse density. And decreased mouse density is leading to decreased prevalence with time delays. But we're also seeing that transmission rate is higher. So I've called that a component amplification effect. So both dilution and amplification are occurring at the same time in the same system. We're just seeing the dilution effect part is stronger. So that leads to the question, what's causing the amplification effect? We couldn't, with this particular study, separate out contact rates and transmissibility. We estimate them together as this transmission rate. And so if we want to know which one is going on, we need to do some experiments. And how might that work? So let's just go through a scenario real quick. So let's say this component amplification effect, could it be through increased contact rates? Well, one way this could happen is here's our back to our hypothetical scenario where we have 3 infected mice in a mouse population. We add some diversity. Perhaps that it changes the behavior of mice in such a way that they're trying to stay away from those dominant competitors. They change their behaviors and kind of congregate more locally in kind of refugia away from these competitors, which could increase their contact rates. So that could be what's happening, and that was going to lead to more infection. Another thing, so the transmissibility side. Another possibility is that with all these competitors around, mice are more stressed. That's stressing their immune system. Their immune system isn't going to work as well, and they're going to be more likely to get infected if they're exposed. So competitors increase stress. So that's my stress metric there, and makes it more likely to get infected because they have depressed immune systems. So that's a possibility. So what I'm working on now is a new project funded by NSF, where we've set up these experimental enclosures. We're going to stock them with different densities of deer mice and with competitors and without competitors, and see what happens. What changes with their contact rates? What does their stress levels look like with and without competitors? What do their immune system functioning look like with and without competitors? So I'll skip past the details so that there's some time for questions. But some of the ways we're going to measure contact rates, potentially with fluorescent powder. You stick fluorescent powder on the animals, you release them, you come back the next day and you put them under UV light and see, did they transfer powder to other animals? We're also messing with potentially with some other cool methods to measure contact rates. So the big picture question are, how do all these things fit together? We're seeing both occurring at the same time in the same system. So this is a cool model system to sort of ask bigger picture questions. When do we expect to see dilution versus amplification? How do species and competitors in particular affect all these different ways things could get transmitted: context, density, stress, immunity, and when would we expect them to act synergistically versus antagonistically? So how does it all scale up? And which anthropogenic stressors are leading to more zoonotic outbreaks? So clearly these zoonotic diseases are important. We've experienced firsthand over the last few years the global impacts they can have. And if we want to prevent future pandemics like this one, we need to understand how all of our actions are affecting all of these different mechanisms of transmission and scaling up. And how can we predict this from happening again in the future? I am ready for any questions.
Thank you, Angie. That was fascinating. And I have a question for you, but I'm going to turn it over to members of the audience first to make sure we get their questions answered. And we'll start with our colleague Curtis here in the School of Public Health. So Curtis, take it away.
Hey, Angie, thanks for sharing that. It was really cool. And I'm looking forward to learning more about your NSF project. I had a question just on that you touched upon at the very end, which had to do with the markers that you're going to use to evaluate stress in these animals. And I'm just curious, like when you're capturing the animals and collecting, a sample, I mean, that's a really stressful, this is probably like, ecology 101 kind of question, I guess, but like how do you assess sort of like longer term or, some sort of integrated measure, time integrated measure of stress instead of just like acutely?
Yeah, so we're mostly using fecal samples. So we're looking at fecal glucocorticoid metabolites. And so they're, metabolized parts of these, stress glucocorticoids. And so they're a longer, more integrated measure than like taking a blood sample. And so we set out traps in the evening, we come back within 4 hours. And so we know that any poop we get from the animal or from the trap is less than 4 hours old. And then we can say that, okay, that shouldn't have been caused from stress-induced traps.
Yeah.
So that's one of the main ways. We're also looking, we're still exploring different measures. We're looking at sort of more integrated, like just neutrophil lymphocyte ratios, body condition, kind of bigger, broader measures. But the fecal measures are a bit better than taking something like from the blood, because for sure you would get stressed animals right then and there. And we're hoping that that's a more integrated measure.
Yeah, great. Thanks, Angie.
Any other questions from the audience? If you want to type in the chat or just unmute yourself on camera or off and ask away.
Dr. Luis, thank you for the presentation. It was very, very interesting. I was wondering, do you have any thoughts about how different predators of deer mice might fit into population density dynamics?
Great question. I wish we knew more. A lot of the data that we've collected doesn't do a great job at that. I'm using a lot of studies, like most of what I told you about was from studies in the Southwest where they were just looking at small mammals, and we don't have a great measure of what's going on with predators there. Around here, we have a little bit more knowledge about what's going on because I have a colleague, John Marin, who is based in EE and just retired. But he had some really great long-term field studies up at Bandy Rancher, which is actually where we're going to do our long-term experiment, where we're going to do our enclosure experiments. He had these great exclosures where he was looking at what happens when you exclude large ungulates or avian or even deer mice. And so he had some great paired experiments where he had these exclosures where they excluded everything but deer mice, which would include the deer mouse predators. So you don't have any mesocarnivores or avian predators that are allowed because they're being kept out in these exclosures, but there's little tiny holes so that deer mice can get in, and voles. And basically what we saw there was that there was no effect of predators on deer mice. There was a huge effect of predators on voles. So that's really interesting given this whole community dynamic thing. Like when you've got predators, it might change the interaction between voles and deer mice because voles are more affected by the predators than the deer mice are. But we're really interested in this, and we're thinking about better ways to try to get at how the predators might be important, but we're not quite there yet.
Thank you.
Any other questions? Angie, I have a big picture question, and this pertains to some of the things that you mentioned at the beginning of your talk, specific to bats in Southeast Asia and the diversity of bat populations there, and the relationship potentially with our recent pandemic and other potential zoonotic disease outbreaks coming down the track. Based on your understanding of the Hantavirus system, do you think that there are any general lessons to be learned about like the increasing pressure of climate change on diversity and or the expansion of the human population in terms of the increased or decreased potentially risk of other zoonotic disease outbreaks in humans? Or do you feel that the specifics of the natural history of any particular either species or disease makes it impossible to do sort of broad strokes?
Yeah, so that's what I'm hoping to get at a bit with this, is to think about like, what are all the different mechanisms? When would we expect to see this thing happening versus this thing, and how might they all fit together? Like, when do they work together and, synergistically versus, trading off? I'm not sure, but given the examples that I've talked about, whether it's dilution or amplification, I mean, the biggest thing is just leave everything alone, leave all these wildlife populations alone, because whether it is us removing and killing animals and decreasing biodiversity, or us going into these biodiverse areas and cutting down habitat and exposing ourselves to these things, like all of those things are definitely leading to these disasters. Like we kind of know that. And so each mechanism might be less important than just having a better strategy to leave these populations intact without the human disturbance and the stress and the contact. Mm-hmm. I don't think that really answered your question. There's been a lot more on the vector side. And so there's been a lot of studies looking at climate and mosquitoes and how we would expect them to spread. And there's a lot of evidence that there's going to be changes in that kind of spread, largely increases in mosquito-borne disease and maybe tick-borne disease as well. I tend to concentrate more on the directly transmitted, and there it can be all over the place. It sort of depends on, climate and particular anthropogenic stressors. But again, like we just need to leave these populations intact and alone, and we'd probably be good.
Yeah. On the public health side, but also medical anthropology as well, we get so many students interested in understanding the relationship between climate change and health, specifically with these kinds of disease outbreaks. Sometimes, not always, but that is a common interest. And so it's just, I think the takeaway from your talk is that specific to the host species, specific to the natural history of the disease in question, there are a lot of different factors that can come into play, and that it seems to me like understanding the specifics of the interplay is the heart of the matter at this point in time in our scientific understing. But it sounds like there's so much work left to do and abundant dissertation topics, exactly. And more exciting stuff in store for you as well. Thank you so much for joining us. We're right at 2:50 now, which is when we generally like to let folks get to their 3 p.m. But if there is another quick question, I think we can maybe take one more. It seems like people are satisfied for now. Angie. So again, thank you so much. I'll just say that next week we're going to be welcoming another very famous medical anthropologist, Mimi Nichter, who is going to be talking to us about her work on tobacco cessation in Indonesia. She'll be introduced by our colleague Gil, who you all saw on the call today, and we have that to look forward to next week. So thanks again for joining us for the series, and to Dr. Luis for being our speaker this week. Thank you.
Thanks, Dr. Luis.
Thanks.